Hello, everyone. So my name is Iram. I am a rising senior majoring in cellular biology, and I work this summer. I worked at the Okuda lab at University of North Carolina, Chapel Hill. And my project involved a lot of experimental and computational work. So I'm going to take you through all of the things I did over the summer. Um, so a little bit of background. So the lab worked, sorry, yeah, the lab worked with cystic fibrosis, and cystic fibrosis is basically a recessive autosomal disorder caused by mutations in the CFTR gene, which is basically affecting the lung tissues, and that would impact the airway hydration and pH regulation, which directly impacts the mucus flow and proper functioning of the lung. And because it's kind of a rare disease, but because it has a genetic cause, what we were trying to look at was CFTR gene. So CFTR plays a very important role in the airway surface liquid hydration. And the genes that express CFTR were previously studied and identified, and one of the main focuses of this research has been understanding what are the different kinds of cells that express this gene and to what extent, so that we can understand what could be cellular targets for CF. And it's been previously identified ionocytes, which is a rare cell type, which is just 1% of human bronchial epithelial cells. They are the one contributing a significant portion of CFTR. However, there are also additional secretory or goblet cells, which do express CFTR, but which is what our lab is studying, and to see what other cell types except for ionocytes are also responsible for CFTR secretion, which could play a role in understanding how CF really functions. And a previous so in this research, we use IL-1 beta, which is basically a cytokine to replicate a CF or an inflammatory conditions in our cell cultures. And we were trying to see how the cells would respond to the IL-1 beta treatment. And currently, a clear mechanism is unknown on how the cells react, or does the CF secretion increase or expression increase or decrease in certain cell types when they are in an inflammatory state compared to a control state. So the research goal of this project was to understand how some specific cell types would have a varied expression with IL-1 beta treatment. And this is just an overview of the human airway epithelial cells, because there are, the further down we go, we could get into the specifics of how many different kinds of epithelial cells there are. But majorly, we looked at the goblet or the secretory cells versus the rare type, which is the ionocyte. And okay. Yeah, and the methods that we used were firstly was experimental, which was a lot of cell culture. So taking the airway epithelial cells, both from the proximal and distal airway, and then culturing them in ALI, which is basically air liquid interface, to replicate how cells grow in vitro to replicate the lung conditions. And then we collected qPCR data as well as single-cell RNA sequence data, which we further used for the bioinformatics analysis. And bioinformatics analysis was basically just running the RNA-seq data to see what certain cell types are present in the samples, and how the treatment how with each treatment the different CFTR mRNA expression was changed. And we did a lot of this through the CREP package on R, and as a lot of research community is aware of, R is a big coding tool to work through big data sets. And we ran, I guess there were 15 samples, which we later used to differentiate into 13 different cell types. So basically, the results of the RNA-seq data showed us that the distribution across the cell types was pretty varied. These are three UMAPs, which basically show how the samples that we got were distributed at a very different level. So like if the first UMAP basically shows the three different donor samples that we use, so these are from three different people and three donors. So their distributions of the cells are like varied and compared to like each other. And the second UMAP is basically the same figure, but like we identified the UMAP based on treatment, which is control versus IL-1 beta, which is to see the inflammatory condition versus like normal conditions in the cell. And the last and the most colorful UMAP is the one with cell types. We identified 13 different cell types through our RNA-seq data to basically show how all of these cell types were forming clusters. By cell clusters, we generally mean cells that show similar properties, so basically one type of cells. And what interesting is that as we can see, like not all the donor distribution, if you look at like maybe the green part in the first result figure, does not align exactly with just one color in the cell type cluster in the third UMAP, which is like interesting because like that also makes us think on like what kind of donor sample is contributing to what kind of cells in the samples that we are analyzing. And after we were able to identify 13 different types of cell types, we basically annotated each cell types based on the marker genes. So we identified top 10 marker genes in each of the clusters, and we did a lot of cell prediction to see what cell cluster would be a specific cell type, such as basal or ciliator or secretory cells. And then manually checked with people who've been working in this, so to see if our predictions make sense with like the data and the marker genes that we have. So here's the annotated marker gene list. And because our focus was to see the CFTR expression in different clusters, when you look at the violin plot right below it, it shows the CFTR expression in both the control and the IL-1 beta in all of these clusters. And what we found interesting was that the CFTR expression actually is decreased in like the 12, in this case, the 13th cluster, which is technically the ionocyte, compared to control, which was not the first result we were predicting. So that led us to like seeing it, which led us to like dividing the data into like the two time slots that we have, which was 6 hours and 72 hours. The data was collected at both of these time slots. So what we did was just to create another violin plots for like different time stops. And we found certain decrease in the CFTR expression in ionocyte after 6 hours. However, we didn't find significant decrease at 72 hours. And when it came to secretory cells, we found significant decrease in CFTR expression, which was not found at 6 hours. And this also is very interesting because of the CFTR mRNA production after a certain period of time changes, and also the cell population and the cell clusters also change with time. So after this, we were not able to have like a conclusive enough analysis onto which cell type is producing how much kind of CFTR. So we then divided or basically analyzed how much of the cell proportion or how much of the cells in each cluster was producing CFTR. And from the bar charts that you see here, it's basically so we figured that in secretory and the ionocytes, the CFTR expression went much higher in 72 hours, which again was interesting because it showed like the proportions of cells that were secreting CFTR, which were much higher at 72 hours. However, there was not significant like increase. Instead, it was like the opposite. So the interest was to look at secretory cells versus ionocytes, and then we basically found qPCR results for to see if the ionocyte number went up, and ASCL3, which is an ionocyte marker, showed us that there was not particularly any increase in the ionocyte cell numbers or markers. So basically, which also showed us that even though the proportion of ionocytes increasing, the proportion of ionocytes that express CFTR do increase, the cell number doesn't really increase. And then we also did qPCR for CFTR and MUC5B, which is for another secretory cell marker, to see if there was any significant increase. And the results aligned with the predictions on like IL-1 beta increase CFTR and MUC5B expression. However, we didn't see any increase in ionocytes. And so basically, a lot of this analysis and a lot of this research and questioning and troubleshooting led us to like certain conclusions of how CFTR expression does not show major change with IL-1 beta treatment. But it would be interesting to look at how certain cell type expressions would be with like would be treated at different time slots. So maybe collecting data at 24 hours, or maybe collecting data from more donors would help us show what if there is any significant increase or decrease in the CFTR expression. And we identified, like with the previous data, that it matched that ionocytes and secretory cells are the major cell types expressing CFTR both before and after the IL-1 beta treatment. And they could be used as like important cellular targets for CFTR correction for potential drugs or like therapies. And ionocyte frequency did not increase with like IL-1 beta treatment. So this also led us to the future direction, which means that when I was trying to talk about the HuBMAPs, or like how certain clusters don't align with the certain cluster code, or certain types of cells are not coming from the same donor. So like some specific cell types are only coming from one donor, or there's like variance when it comes to donor samples. So what would be interesting is to increase the donor samples so that we have more generalizable results and collect the data at different times to see how CFTR expression is different in both normal and CF airway or inflammatory conditions. And then further categorize how like cell type specific CFTR function in airway epithelial would be different, because like we only looked at the expression for this project, but however, like looking into function would also give us like more deeper insight into how certain cells that produce a specific type or a specific amount of CFTR. And this, I would like to thank all of my lab members and people who helped me with experimental and computational work and helping with all the setup. And it's been honestly such a learning experience to work in this research project, because it involved so much collaboration and support from so many people. And I've learned a lot over the summer, and it's been like incredible to just see how like tiny and small things could affect big pictures, how certain gene expression could be important or interesting to look at, and how certain airway functions are determined by certain gene expressions. And here are a few of the references that I could share, and I would be happy to answer any questions if there are any.